Citric acid cycle.
The citric acid cycle, also known as the tricarboxylic acid cycle, or the Krebs cycle, is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate derived from carbohydrates, fats and proteins into carbon dioxide and chemical energy in the form of adenosine triphosphate. In addition, the cycle provides precursors of certain amino acids as well as the reducing agent NADH that is used in numerous other biochemical reactions. Its central importance to many biochemical pathways suggests that it was one of the earliest established components of cellular metabolism and may have originated abiogenically. The name of this metabolic pathway is derived from citric acid, a type of tricarboxylic acid, that is consumed and then regenerated by this sequence of reactions to complete the cycle. In addition, the cycle consumes acetate, in the form of acetyl-CoA, and water, reduces NAD+ to NADH, and produces carbon dioxide as a waste byproduct. The NADH generated by the TCA cycle is fed into the oxidative phosphorylation, electron transport, pathway. The net result of these two closely linked pathways is the oxidation of nutrients to produce usable chemical energy in the form of ATP. In eukaryotic cells, the citric acid cycle occurs in the matrix of the mitochondrion. In prokaryotic cells, such as bacteria which lack mitochondria, the TCA reaction sequence is performed in the cytosol with the proton gradient for ATP production being across the cell's surface, plasma membrane, rather than the inner membrane of the mitochondrion. Several of the components and reactions of the citric acid cycle were established in the 1930s by the research of the Nobel laureate Albert Szent-Györgyi, for which he received the Nobel Prize in 1937 for his discoveries pertaining to fumaric acid, a key component of the cycle. The citric acid cycle itself was finally identified in 1937 by Hans Adolf Krebs while at the University of Sheffield, for which he received the Nobel Prize for Physiology or Medicine in 1953.
Evolution.
Components of the TCA cycle were derived from anaerobic bacteria, and the TCA cycle itself may have evolved more than once. Theoretically, there are several alternatives to the TCA cycle. However, the TCA cycle appears to be the most efficient. If several TCA alternatives had evolved independently, they all appear to have converged to the TCA cycle.
Overview.
The citric acid cycle is a key component of the metabolic pathway by which all aerobic organisms generate energy. Through catabolism of sugars, fats, and proteins, a 2-carbon organic product acetate in the form of acetyl-CoA is produced. Acetyl-CoA along with 2 equivalents of water, H2O, is consumed by the citric acid cycle producing 2 equivalents of carbon dioxide, CO2, and 1 equivalent of HS-CoA. In addition, 1 complete turn of the cycle converts 3 equivalents of nicotinamide adenine dinucleotide, NAD+, into 3 equivalents of reduced NAD+, NADH, 1 equivalent of ubiquinone, Q, into 1 equivalent of reduced ubiquinone, QH2, and 1 equivalent each of guanosine diphosphate, GDP, and inorganic phosphate, Pi, into 1 equivalent of guanosine triphosphate, GTP. The NADH and QH2 generated by the citric acid cycle are in turn used by the oxidative phosphorylation pathway to generate energy-rich adenosine triphosphate, ATP. One of the primary sources of acetyl-CoA is sugars that are broken down by glycolysis to produce pyruvate that in turn is decarboxylated by the enzyme pyruvate dehydrogenase generating acetyl-CoA according to the following reaction scheme. CH3C equals O, C equals O, O, pyruvate, plus HSCoA plus NAD plus CH3C, equals O, SCoA, acetyl-CoA, plus NADH plus CO2. The product of this reaction, acetyl-CoA, is the starting point for the citric acid cycle. Below is a schematic outline of the cycle. The citric acid cycle begins with the transfer of a 2-carbon acetyl group from acetyl-CoA to the 4-carbon acceptor compound, oxaloacetate, to form a 6-carbon compound, citrate. The citrate then goes through a series of chemical transformations, losing two carboxyl groups as CO2. The carbons lost as CO2 originate from what was oxaloacetate, not directly from acetyl-CoA. The carbons donated by acetyl-CoA become part of the oxaloacetate carbon backbone after the first turn of the citric acid cycle. Loss of the acetyl-CoA donated carbons as CO2 requires several turns of the citric acid cycle. However, because of the role of the citric acid cycle in anabolism, they might not be lost, since many TCA cycle intermediates are also used as precursors for the biosynthesis of other molecules. Most of the energy made available by the oxidative steps of the cycle is transferred as energy-rich electrons to NAD+, forming NADH. For each acetyl group that enters the citric acid cycle, 3 molecules of NADH are produced. Electrons are also transferred to the electron acceptor Q, forming QH2. At the end of each cycle, the 4-carbon oxaloacetate has been regenerated, and the cycle continues.
Steps.
2 carbon atoms are oxidized to CO2, the energy from these reactions being transferred to other metabolic processes by GTP, or ATP, and as electrons in NADH and QH2. The NADH generated in the TCA cycle may later donate its electrons in oxidative phosphorylation to drive ATP synthesis. FADH2 is covalently attached to succinate dehydrogenase, an enzyme functioning both in the TCA cycle and the mitochondrial electron transport chain in oxidative phosphorylation. FADH2, therefore, facilitates transfer of electrons to coenzyme Q, which is the final electron acceptor of the reaction catalyzed by the succinate, ubiquinone oxidorductase complex, also acting as an intermediate in the electron transport chain. The citric acid cycle is continuously supplied with new carbon in the form of acetyl-CoA, entering at step 1 below. Mitochondria in animals, including humans, possess 2 succinyl-CoA synthetases, one that produces GTP from GDP, and another that produces ATP from ADP. Plants have the type that produces ATP, ADP forming succinyl-CoA synthetase. Several of the enzymes in the cycle may be loosely associated in a mulch-enzyme protein complex within the mitochondrial matrix. The GTP that is formed by GDP forming succinyl-CoA synthetase may be utilized by nucleoside diphosphate kinase to form ATP. The catalyzed reaction is GTP plus ADP, GDP plus ATP.
Products.
Products of the first turn of the cycle are 1 GTP, or ATP, 3 NADH, 1 QH2, 2 CO2. Because 2 acetyl-CoA molecules are produced from each glucose molecule, 2 cycles are required per glucose molecule. Therefore, at the end of 2 cycles, the products are 2 GTP, 6 NADH, 2 QH2, and 4 CO2. The above reactions are balanced if Pi represents the H2PO4 ion, ADP and GDP the ADP2 and GDP2 ions, respectively, and ATP and GTP the ATP3 and GTP3 ions, respectively. The total number of ATP obtained after complete oxidation of 1-glucose in glycolysis, citric acid cycle, and oxidative phosphorylation is estimated to be between 30 and 38.
Efficiency.
The theoretical maximum yield of ATP through oxidation of 1 molecule of glucose in glycolysis, citric acid cycle, and oxidative phosphorylation is 38, assuming 3 molar equivalents of ATP per equivalent NADH and 2 ATP per FADH2. In eukaryotes, 2 equivalents of NADH are generated in glycolysis, which takes place in the cytoplasm. Transport of these 2 equivalents into the mitochondria consumes 2 equivalents of ATP, thus reducing the net production of ATP to 36. Furthermore, Inefficiencies in oxidative phosphorylation due to leakage of protons across of the mitochondrial membrane and slippage of the ATP synthase proton pump commonly reduces the ATP yield from NADH and FADH2 to less than the theoretical maximum yield. The observed yields are, therefore, closer to 2.5 ATP per NADH and 1.5 ATP per FADH2, further reducing the total net production of ATP to approximately 30. An assessment of the total ATP yield with newly revised proton to ATP ratios provides an estimate of 29.85 ATP per glucose molecule. However, the total amount of ATP generated from just the Krebs cycle for one molecule of glucose is around 25.
Regulation.
The regulation of the TCA cycle is largely determined by product inhibition and substrate availability. If the cycle were permitted to run unchecked, large amounts of metabolic energy could be wasted in overproduction of reduced coenzymes such as NADH and ATP. The major eventual substrate of the cycle is ADP, which gets converted to ATP. A reduced amount of ADP causes accumulation of precursor NADH, which in turn can inhibit a number of enzymes. NADH, a product of all dehydrogenases in the TCA cycle with the exception of succinate dehydrogenase, inhibits pyruvate dehydrogenase, isocitrate dehydrogenase, a ketoglytarate dehydrogenase, and also citrate synthase. Acetyl-CoA inhibits pyruvate dehydrogenase, while succinyl-CoA inhibits alpha-ketoglytarate dehydrogenase and citrate synthase. When tested in vitro with TCA enzymes, ATP inhibits citrate synthase and a ketoglytarate dehydrogenase. However, ATP levels do not change more than 10% in vivo between rest and vigorous exercise. There is no known allostric mechanism that can account for large changes in reaction rate from an allostric effector whose concentration changes less than 10%. Calcium is used as a regulator. Mitochondrial matrix calcium levels can reach the tens of micromolar levels during cellular activation. It activates pyruvate dehydrogenase phosphatase, which in turn activates the pyruvate dehydrogenase complex. Calcium also activates isocitrate dehydrogenase and a ketoglytarate dehydrogenase. This increases the reaction rate of many of the steps in the cycle, and therefore increases flux throughout the pathway. Citrate is used for feedback inhibition, as it inhibits phosphofructokinase, an enzyme involved in glycolysis that catalyzes formation of fructose 1,6-bisphosphate, a precursor of pyruvate. This prevents a constant high rate of flux when there is an accumulation of citrate and a decrease in substrate for the enzyme. Recent work has demonstrated an important link between intermediates of the citric acid cycle and the regulation of hypoxia-inducible factors, HIF. HIF plays a role in the regulation of oxygen homeostasis, and is a transcription factor that targets angiogenesis, vascular remodeling, glucose utilization, ion transport and apoptosis. HIF is synthesized constitutively, and hydroxylation of at least one of two critical proline residues mediates their interaction with the von Hippel-Lindau E3 ubiquitin ligase complex, which targets them for rapid degradation. This reaction is catalyzed by pronyl 4-hydroxylases. Fumarate and succinate have been identified as potent inhibitors of pronyl hydroxylases, thus leading to the stabilization of HIF.
Major metabolic pathways converging on the TCA cycle.
Several catabolic pathways converge on the TCA cycle. Reactions that form intermediates of the TCA cycle in order to replenish them, especially during the scarcity of the intermediates, are called anaplerotic reactions. The citric acid cycle is the third step in carbohydrate catabolism, the breakdown of sugars. Glycolysis breaks glucose, a 6-carbon molecule, down into pyruvate, a 3-carbon molecule. In eukaryotes, pyruvate moves into the mitochondria. It is converted into acetyl-CoA by decarboxylation and enters the citric acid cycle. In protein catabolism, proteins are broken down by proteases into their constituent amino acids. The carbon backbone of these amino acids can become a source of energy by being converted to acetyl-CoA and entering into the citric acid cycle. In fat catabolism, triglycerides are hydrolyzed to break them into fatty acids and glycerol. In the liver, the glycerol can be converted into glucose via dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by way of gluconeogenesis. In many tissues, especially heart tissue, fatty acids are broken down through a process known as beta-oxidation, which results in acetyl-CoA, which can be used in the citric acid cycle. Beta-oxidation of fatty acids with an odd number of methylene bridges produces propionyl-CoA, which is then converted into succinyl-CoA and fed into the citric acid cycle. The total energy gained from the complete breakdown of one molecule of glucose by glycolysis, the citric acid cycle, and oxidative phosphorylation equals about 30 ATP molecules, in eukaryotes. The citric acid cycle is called an amphibolic pathway because it participates in both catabolism and anabolism.
Interactive pathway map.
Click on genes, proteins and metabolites below to link to respective articles. The interactive pathway map can be edited at Key Pathways, TCA cycle WP70.